In our second module of protein macromolecule interactions, we will be looking at protein-protein interactions, protein-peptide interactions, and a discussion on chaperone proteins and what we call IDPs, intrinsically disordered proteins. In the last module, we looked at protein carbohydrate and protein nucleic acid interactions. In each of these cases, we are looking at a ligand interacting with the protein. The ligand in this case is the carbohydrate, the nucleic acid, and in the next two lectures, we will be looking at proteins as the ligands. The interactions of proteins are extremely important, as we have seen, in several aspects of biochemical processes. What we are going to look at is the types of protein-protein interfaces, the properties of these interfaces, and specific binding affinities related to the protein-ligand interactions that we had studied before. In an understanding of what we mean by protein-protein interactions that are commonly called PPIs, these are a fundamental part of all major biological processes, as we have seen in our lectures. They occur when two or more proteins bind together in a specific manner, and there are many functions that are regulated and mediated and even controlled by this complex formation, and almost all biological activities of cells occur through these interactions. Inside the cell, we have several complexes that are formed to participate in molecular processes, as we have seen, that are built up through these PPIs. So, a knowledge of these PPIs can actually improve our understanding of diseases to help design and develop therapeutic approaches. And we will see how this specific topic can be approached from an understanding of the protein-protein interactions. The specific functions, therefore, are influenced by the structure of the protein and the interaction type interactions that occur. So if we look at protein-protein interactions in general, they can be of two types. We can have permanent complexes where the interactions are strong and irreversible. For example, this could be a case where we have monomeric subunits of a protein forming an oligomeric unit, where the interactions are strong, they are irreversible in a way, and they form a permanent complex. On the other hand, we could have what is called a transient complex, where there is a specific change in the oligomeric state of the protein, or it could just interact for the specific process or interaction or reaction to occur. These can be of the weak type, where we could have a dynamic mixture of different states, or we could have a strong type where there is a transition from an unbound or a weakly bound to a strongly bound state upon triggering. That could be the binding of a small molecule, a metal, or a cofactor that is going to trigger a specific reaction that requires a protein-protein complex to be formed. If we look at the affinity, or rather the dissociation constant values that we had studied previously in protein ligand interactions, we realize that the binding affinity is going to be very strong for a permanent complex, as reflected in the values of the Kd constant. We know that a smaller Kd indicates a tighter binding, and the weak are of the order of micromolar values for the Kd. So the protein-protein interactions can occur in multi-subunit proteins, say for example hemoglobin, the ribosome, RNA polymerases. We have transient protein interactions such as protein kinases, phosphatases, and what they do is they interact in a very transient manner with the specific protein that it has to complex with, and then performs the particular reaction or the specific biochemical process that has to occur, and that would be a transient type of interactions. There are some stable interactions as well that are long-term interactions that have specific functional or structural roles. The examples that we have looked at or we have studied in the process of this course is, for example, electron transfer proteins. Here, the interactions among the proteins are very dependent on a highly specific binding between the proteins so that there is efficient electron transfer in the electron transport chain that we have looked at previously. For signal transduction, where we need signal propagation, this also depends highly on the protein-protein interactions between the various signaling molecules in the cells. In addition, we have membrane transport, the transport of proteins by another protein from the cytoplasm to the nucleus, or vice versa, where we have a specific integral membrane protein or a peripheral membrane protein that would take part in this type of transport. The cell metabolism, where we have the interactions between enzymes that are involved in bioprocesses that produce the small compounds or other macromolecules in a specific enzymatic type of reaction. Then muscle contraction, where we looked at motor proteins, where we have the motor protein involvement, for example, the myosin and the actin that have to act together in their molecular motor action. So the types of protein-protein interfaces that we have here can be of a functional type. They could be enzyme, substrate, antibody, antigen, receptor, hormone, or signal transduction type. There can be a similarity of the binding partners. They could be similar, which could be in a homotype or a heterotype. And the type of binding can be transient or permanent. So the interactions that occur in these specific types of protein-protein interfaces, they could include electrostatic forces, hydrogen bonding, hydrophobic effect, similar to what we see in the specific types of interactions that we saw in tertiary structure, the non-covalent types of interactions. The amino acid composition that is seen at the protein-protein interface is 50% polar in nature approximately, in the middle between the protein surface and the core. And the polar residues in the interface are usually uncharged in this case. And we see that they are richer in aromatic residues and histidine than the protein surface. What we have something called hub proteins, which we will see what it means in a moment, where there is a binding with different partners. So if we look at the properties of the interface, it is important to consider the type of complex when we are characterizing this protein-protein interaction interface. For example, if we're looking at a trypsin pancreatic trypsin inhibitor complex, then there are specific residues that are involved in the interaction. So we have the free pancreatic trypsin inhibitor here. These are the specific residues that are involved in the interaction. The characteristics of these residues is such that they would have an interacting partner in the partner protein that it is interacting with. In this case, we have a lysine, which has an electrostatic interaction with an aspartic acid of the protein. Then we look at the specific types of interactions. So these types of interactions, as we saw, could be an electrostatic type of interaction, which we usually see on the surface of a protein, and they could be of a hydrogen bonding type, disulfide bonds that would connect two parts of a protein together, could be of an intermolecular type, and we have ring stacking that we would see for the aromatic interface. So the properties that we see for a non-covalent tertiary structure interaction and the covalent interactions that we observe are similar to what we can see in the different types of proteins where we have a homo complex or a hetero complex. So when we study this molecular structure, we can get details about the interface. And this study or this understanding enables us to understand the interaction between the proteins. The parameters here are the size. What is the solvent accessible surface area? We have looked at this previously, the topic of accessible surface area. How much of the protein molecule is accessible for interaction with another molecule. The shape of the molecule, the complementarity between the surfaces. We need a geometric complementarity as well as a chemical complementarity. The residue interface propensities, the hydrophobicity, and the secondary and domain structure that is also going to be important in an understanding of what triggers a protein-protein interaction to occur and the conformational changes that might occur due to complex formation. What we have is an average of 77%, approximately 77% of inter-subunit hydrogen bonds that are found between side chains. However, an average of about 68% of the hydrogen bonds are found within subunits are between the backbone atoms. So if we look at the hydrogen bonds in the backbone and the side chains, we have approximately a 77% that are inter-subunit type. In contrast, we have 68% that are within the backbone atoms here. So the secondary structure elements that are considered here are usually not continued over subunit boundaries, so they are confined within the specific subunit that they occur in. So when we're looking at the non-polar interactions, we have the affinity. The electrostatic interactions bring about some specificity. And both of these are important in the complex formation that we see that are going to bring these two protein molecules together. So the non-polar interactions are going to get the affinity and the specificity, both of which are important in bringing of the formation of a protein-protein complex that is going to be a result of protein-protein interactions. The geometrical properties that we have to look at is the specific types of surface area. The surface area can range between 1000 angstrom squares to even 15,000 angstrom square, covering 5 to 75 residues in the protein. And the interfaces can vary in their geometric complementarity. So if we look at the specific types, they can be homodimers. These are frequent, permanent, obligatory complexes. That means they have an obligation to interact with each other. And heterotrimers are often transient. For example, if we're looking at an enzyme inhibitor type or a receptor hormone type of complex. So if we look at the specific type of temporary complexes that may occur, or we can have permanent complexes. The types of protein interfaces are going to depend upon the interactions present and the chemical complementarity that we look at. So we can have an example of a temporary complex where we have different types of units. We can also have an obligatory one, that is a subunit type, that are obligated to stay together in the dimeric form of the protein. So when we look at obligatory homomers versus non-obligatory homomers, the interfaces tend to be larger, more hydrophobic, more complementary, and have a greater affinity because they are obligated to stay together to form the stronger complex that we see in dimeric proteins or in specific oligomeric proteins. And when we look at the specific induced fit upon complex formation, this means the induced fit means that the molecules adapt to interact with the other subunit so that they form this complex. So when we look at the electrostatics of the interactions, the electrostatic complementarity can vary greatly. We have polar patches on the surface. They form interaction networks that reduce the destabilization due to desolvation. We have to remember that when we have the protein molecule is surrounded by the water molecules that solvate the protein molecule. Now, if we have another protein molecule that is going to come to interact with this, the interface water residues are destabilized due to the desolvation process when we have the complex formation. So the interaction network could involve polar patches. We will also see how we have some water molecules that can also be present in the interface. They have positively charged residues that are comparatively more arginine than lysine. And we have the complementarity of the surfaces here that would result in a strong interaction given that we know that we have these are the negative the negative patches, and these are the positive patches. So depending upon the structural electrostatic complementarity, we can have a complex formation. So we have the positive and negative parts here. So the protein-protein interface can also undergo conformational changes. The induced fit that occurs is usually small, and this is restricted to the interfacial side chains that accommodate to form the complex formation. So what happens is this makes it easier for the two proteins that are coming together to bind at a lower entropic cost. The binding affinity that we see in the specific types of examples given here are for signaling and electron transfer. In this case, proteins tend to have a lower affinity than average because it is required for quick binding and release. So you do not want too tight a complex to form. So the affinity is not that high. The cell-cell recognition process also have to have lower affinity than the receptor hormone complexes, and the immune response, however, has increased affinity of the antibodies to their specific ligands or the antibody-antigen interactions. So the affinity that we see results from a small number of residues that are confined on the surface that are termed hotspots. These hotspots are involved in the specific interaction that is going to result in the interface residues interacting with each other to form the protein-protein complex in the protein-protein interaction. So the binding hotspot that we see in this particular example, where we have the lactate dehydrogenase interface, indicates that in chain A and chain C, there are specific binding hotspots that actually appear as clusters on the surface. And these hotspots contribute to the overall delta delta G of the binding free energy. And if we look at the two complexes in this form, where we have an open sandwich view of the same interface, where the two subunits are just rotated 90 degrees to each other. So this is where we have the interacting hotspot that is important in bringing these two chains together to form the specific complex. So when we look at these hotspots, they tend to be conserved in nature and they appear in tightly packed regions of the interface. They appear in clusters and they have specific hotspot clusters in specific proteins that are the interface, that are the result of this specific interaction to occur, where in this case, this is an example of a hotspot cluster between the beta lactamase and the beta lactamase inhibitor protein. So these hotspots would result in a strong interaction that would hold the complex together. Again, it depends upon the type of affinity, whether it is low affinity or high affinity would depend upon the type of interaction or the type of specific process that it goes through. So we have in these hotspots a prevalence of aromatic residues as well as histidine, leucine, isoleucine, methionine, and arginine. We have here the PP interfaces, the protein-protein interface, where we have aromatic residues, where we can have an involvement of stacking interactions in the middle of this interface. So these numerous studies show that these aberrant protein-protein interactions can be linked with various diseases, because we understand that the protein-protein complex formation is involved in many biological processes, whether it's signal transaction or cell-cell in addition or even motor protein myosin acting interactions. So targeting these PPIs in the treatment of diseases is considered an essential strategy for the development of new drugs. And there can be modulation of these PPIs that can be recognized for drug discovery. So if we look at the protein-protein interface for drug design, we know that there are interacting units, and the interacting units form specific complexes to get to their functional complexes or their functional forms. So this is where we lose a specific surface area. We know, or we can find out what the specific interface residues are, the hotspots that are going to be involved in the interaction, and we can target the specific points to develop drugs for inhibition of the protein-protein complex formation that would be required to inhibit a specific biochemical biological process. If we look at some specific examples now, we have a family of proteins called the RAS proteins. They are from the rat sarcoma virus. They belong to a class of GTPase proteins. So this RAS is a member of the small GTP binding proteins of the G protein superfamily. This protein is a major regulator of cell growth and development, and oncogenic mutants of RAS, they induce specific transformations in cells. So the roles of this specific protein are in transmitting signals within the cells in what we call cellular signal transduction. The switching on of this RAS protein results in the signals to genes that are involved in cell growth, differentiation, and overall survival. Now, what happens is if there are mutations in these genes, this can lead to a permanent switching on. And what that is going to do, that is going to affect the cell growth and differentiation. So, even in the absence of incoming signals, if these mutations results in an on signal all the time, then the process is going to continue. That is going to be detrimental to health. Now, what happens is if these can interact with specific downstream effectors, the RAS protein signals are involved in a number of distinct pathways. The effectors initiate signal transduction reactions that are required for the biological activity of the RAS. For example, we will just look at an example of an interface. This guanidine nucleotide dissociation stimulator protein specific for RAS is one of the effectors. So the study of these complex interfaces is going to give us some information about how they can be targeted for design of any drugs. So when we are looking at the RAS binding domain of its effector, we are looking at the interface. The complex interface gives us an important platform to study and to analyze the function of the protein and to know where to target it to stop any aberrant protein-protein interactions that is going to lead to disease. So the structure of these bound complexes, bound to the mutants even, can also provide crucial information required for the development of drugs against this RAS protein. So if we look at the specific interface where we have the RAS protein and the RGS-RBD protein type here. So the interaction between the RAS and the RGS-RBD is between the first two beta strands and the long alpha helix of the RBD. So here we have the beta strands and we have the specific, this is the RAS binding domain where we have the interactions. And what we see is the surface are involved in they have hydrophilic side chains that are involved in the interactions. So we understand that the surface properties of the individual proteins as they were in solution would still have the hydrophilic surface or the hydrophilic side chains on the surface of these two proteins, but they are also involved in the interaction between these two proteins. This means that there is going to be some interfacial water because what we see is this is common. About 30% of the interface region can be involved with water. So what we see is because there are hydrophilic residues on the surface that would be solvated, the interfacial water also remains when we have the caged water molecules or the red spheres that are trapped in the protein-protein interface. So we have an apoprotein where we have this specific interfacial water. This participates in hydrogen bonding networks and helps minimize unfavorable desolvation effects. So This is where we have a lot of interaction, where we have hydrogen bonds that involve some of the caged water molecules that are also very important in the protein-protein interaction that occurs. Another very important example is the antibody antigen type of example. The antibody, we know, is a large Y-shaped protein that is used for the immune system to identify and neutralize foreign intruders, be they bacteria or viruses. And we have the antigen, that is a molecular structure or a unique molecule present on the outside of the pathogen that can be bound by an antigen specific antibody. And this association is very important. If we look at an overall structure of the antibody, what we have is we have in the blood the antigens that are specifically bound to antibodies in very high affinity complexes to form the antibody antigen complex. And this immune complex is transported to the cellular systems where it can be destroyed or deactivated. These antigens are bound to antibodies through weak and non-covalent interactions, like we saw for the previous cases: hydrogen bonds, electrostatic interactions, van der Waals forces, and hydrophobic interactions. But these are extremely important interactions in the protein factor. So we have the antibody, we have the light chain, we have what is called an FAB domain, a heavy chain, and we have an antigen binding site. These are very specific in nature with strong, high and very high binding affinity. So when we have the antigen, we have a specific antigen binding site that is going to bind to the antibody. Now, there are lots of variations that can occur in an antibody antigen complex. So, the human antibody consists of 4 polypeptide chains. There are 2 identical heavy chains and 2 identical light chains that connect through disulfide bonds. We have the FAB, that is the fragment, that is the antigen binding fragment that is formed from the amino terminal, as we saw in the previous figure. This region is called the variable domain, and it is composed of amino acid sequences that define each type of antibody and their specific binding to an antigen. So when we look at the binding affinity, we can have increased affinity up to 1500 fold, and mutations can actually indirectly, via their conformational changes, can affect the binding. So we can have a high affinity binding or a low affinity binding depending upon what happens in the variable and the constant regions in their interactions in the antibody, in the binding domains that we see. So if we look at the specific light chain binding here, where we have an arginine 96, we can see that there is the antigen that interacts with the antibody, and there is a specific movement where we would have a low affinity type and a high affinity type that is going to have the specific interaction between this side chain in this specific case. So that is going to be a specific heavy chain that is going to again interact with its specific partner in a high affinity set. So there is going to be a movement of these chains for a facilitated protein-protein interaction. So if we look at the binding affinity, antigens and antibodies show very high binding affinity, like a lock and key interaction. And we have a dynamic equilibrium similarly to what we saw in a protein ligand binding. Here we have an antibody antigen interaction. Again, we can have an equilibrium association constant that is going to give us, from the ratio of K on and K off, going to give us an association constant from which we can understand the specificity and the affinity of this, and this makes it an important tool in experimental biological and research for diagnostics and therapy. This is where we have monoclonal antibodies that act as modulators for targeting these protein-protein interfaces. They compete with PPIs because of their large molecular weight, and the application of these monoclonal antibodies is limited to the extracellular targets. What we have here is some specific type of antibodies that have actually been used in clinical treatment. In this case, this is used in specific types of diseases where the detailed structural characterization of the monoclonal antibodies is required for specific types of interactions with antigens. So what we have, there is in this therapeutic strategy mixture of antibodies that are going to actually interact with specific antigens. And what happens is they have protein-protein interactions that occur, and these can be studied through several methods. So the idea here is that if we have a foreign body, an antigen, we have many antibodies, different types of antibodies that are formed, that are designed to interact or bind with these antigens in the forming of the antigen-antibody complex that then can be destroyed. For example, if we look at an inhibitor of a CD40-CD40L interaction, this is one of the most important receptor-ligand interactions that occurs due to a specific immune response. They participate in several reactions and physiological processes, and the abnormal expression is related to many diseases. So the idea here is that if we block the interaction between the CD40 and the CD40L, it can treat the associated diseases. There is a specific type of anti-CD40 monoclonal antibody that actually blocks the interaction of this CD40 to CD40L, and this has been tested in clinical trials. So what we have looked at is we have looked at the types of interfaces, the protein-protein interfaces, what comprise the interfaces, what residues are likely to be in the interface, what accessible surface area we might lose, and in addition to the geometric complementarity and the chemical complementarity, the types of water molecules that may be present that are trapped in the hydrophilic surfaces of the protein-protein complexes that nevertheless are required, that could be of a permanent type or a transient type. So depending upon the type of protein-protein interface, we would have specific hotspots that would result in a strong affinity that would be required for specific types of protein-protein complexes. These are the references. Thank you.